All right, so we're going to collect data on the part 2, part 3, and part 4 of the procedure of the enzyme lab. So the first one that we're going to go over today after we already did the positive and negative controls before is the enzyme specificity procedure. And so let me screen share so you can see it. And as you can see, part B is enzyme specificity. So there's 3 clean test tubes, and one of them is going to have amylase and water, one of them is going to have amylase and oil, and then one of them is going to have amylase and starch. And based on the positive and negative controls, we already know that amylase and starch produces maltose, which is a sugar, right? And the Benedict's test is what shows us that there was sugar present at the end or not. And so we are going to do these 3 in test tubes 4, 5, and 6. And you're going to basically look at their color after you do the Benedict's test. And you want to remember that the beginning of the Benedict's test is that the solution is a bright blue color. And then after you put it into one of these test tubes and then heat it up, it's going to turn bright orange or brown if there is sugars present. So let's look at the colors for test tube number 4, which is amylase and water. Let me full screen myself again so that you can actually see. So test tube number 4, amylase and water, is this nice bright orange color. So you want to record that down, and then I want you to tell me essentially what does that mean. So you're going to be saying in your table, right, you're going to write the color down, and then you're going to tell me, is there sugar present based on the color change, if there's a color change. And then in conclusion, did amylase break something down? Or if it didn't break something down, what is present and why is it there? Okay, we're going to do the same things for the other 2. So now we're going to go and look at test tube number 5, which is amylase plus oil. And if you look here, what color is the bottom? It's still this bluish color. It's kind of green actually, because the amylase originally is orange. But if you look at the top section right here, that's where the oil is, right? They don't even mix because the Benedict solution is a water and alcohol-based solution. And then oil obviously is hydrophobic, so water and oil don't mix to begin with. And the amylase is stuck at the bottom here with the Benedict solution. And it's still blue because was there any sugar? I don't know, you tell me. The third one that we are going to do is test tube number 6, which is amylase plus starch. And that's our good old number 6 color result. Can you tell me what color it is? Again, remember it started out as blue. You want to tell me, based on the color change, what happened and what is the conclusion that you can explain about the reaction in this test tube. Okay, so this is part B on enzyme specificity. We are basically asking you to compare if the enzyme amylase works on just starch or if it works on other things too. So we also tested it out on oils. So afterwards, we're going to go ahead and answer the questions underneath the table. This is the same table as before. And you're going to answer the question, upon which substrate does salivary amylase work best and why? So you're telling me, does it work best on water? Does it work best on oil? Or does it work best on starch? And keep in mind, the reason why there's a color change here might be due to other factors. So, because remember, water isn't actually a carbohydrate, so amylase technically isn't acting on it. So you can explain the best of your abilities in the conclusion, or you can just ignore this fact, because it was supposed to be a control.